Chapter 17, Protoplasmic Activity. In the last chapter, one type of protoplasmic activity was discussed, and it was shown that the division of the cell depended on colloidal changes in the protoplasm. Beyond any question, many other types of protoplasmic activity also involve colloidal change. The prevention of colloidal change inhibits the possibility of activity. The protoplasm may be kept either in a constant fluid state or it may be kept in a relatively stiff and gel-like condition. In either case, the normal protoplasmic activity is suspended, and if this prevention of activity or of some phase of activity does not result in the death of the protoplasm, it is said to evolve a condition of anesthesia. Here, from a colloid chemical standpoint, it seems probable that anesthesia is really due to the fact that the protoplasm is kept either very fluid or in a highly viscous state, compare Heilbrunn. If the prevention of activity is irreversible, that is to say, if the protoplasm is unable to recover, it is by definition dead. In general, only those agents which cause an increased viscosity or a coagulation can produce an effect which is irreversible. At any rate, no case is known in which an increased fluidity of the protoplasm is irreversible. We are thus led to the conclusion that death is the result of an irreversible coagulation of the protoplasm. This is by no means a new opinion. Indeed, many authors have expressed the view that death is due to an irreversible coagulation of the protoplasm. There are many different types of protoplasmic activity. An amoeba projects pseudopodia and moves from place to place. Other protozoa travel by means of flagella. In plants, there is typically a response to light, to gravity, and to various other factors of the environment. Blue-green algae show peculiar movements which apparently depend on secretion phenomena. There are numerous examples of chemical activity. Gland cells secrete an endless variety of chemical material. Muscle cells contract and impulses travel along nerves. But if we examine more closely, if we study carefully the facts at our disposal, it becomes apparent there are certain phenomena which have much in common. Many types of protoplasm are thrown into sudden activity, or at least react quickly to various agents such as an electric current or mechanical pressure. And the response, wherever it is found, follows the same general laws. It is hardly our purpose to enter a general discussion of the facts regarding irritability. These facts are too well known, and there are too many of them. If we embarked into a complete discussion, we might very well lose track of one or two points it is our desire to bring forward. We shall endeavor to state the most important points as briefly as possible. If in this discussion we refer to facts that are widely unknown, this need not weaken the force of the argument. In the first place, if we are to speak of protoplasmic activity, it might be well to consider what is meant by the resting state. For the purposes of the present argument, let us define the resting state as the state in which the protoplasm normally continues for long periods. This certainly is not an ample definition for all protoplasm, or even for any type of protoplasm under all conditions. But for us in this chapter, it will prove sufficient. For the purposes of our discussion, it will be necessary to consider streaming protoplasm as in a state of rest, in spite of the fact that it is continually in motion. This is certainly a contradiction in terms, but the trouble lies only in the words. The concept is clear enough. The normal state for some protoplasm is a state of slow motion. It remains in this state for long periods, and as far as is known, in many cases at least, no sudden or special changes in the environment exterior to the cell are necessary to maintain this slow motion. Consider now the effect of mechanical pressure or of an electric current. Immediately the motion of the streaming J protoplasm is stopped. See P125 and P132. Moreover, the cessation of movement is not limited to the cell immediately treated, or so to say stimulated. Cessation of movement travels as a wave from cell to cell. Something has happened both to the protoplasm of the cell immediately affected by the physical agent, and also to the protoplasm of the neighboring cells. And that's something we chose to call activity or excitation, in spite of the fact that either of these terms, in some instances at least, is a misnomer. It would be better if we could use some algebraic symbol. Practically every other biological term that we might use has various connotations which make it just as unsatisfactory. We must therefore divorce ourselves from the uncertainties of word disagreements and decide to call activity of streaming plant protoplasm the state in which movement ceases. This state is produced by the electric current and by mechanical pressure. It is also produced in various other ways, but as has already been stated, we shall avoid completeness in order to make our argument more forceful, even if less extensive. The activity or excitation induced by the electric current or by mechanical pressure is prevented by anesthetics. In solutions of ether of about I to 2%, there is no longer any effect of pressure, and no longer any effect of the electric current, compare law about 24. In nerve and muscle cells, the living substance reacts to exactly the same agents that the islet protoplasm does. When nerve and MXXL cells are exposed to the action of an electric current, they are thrown into a state of activity. The muscle cell contracts and an impulse passes along the nerve fiber. Again, both in the case of the muscle cell and the nerve cell, activity is prevented by the addition of ether in approximately 1 to 2% concentration. There is thus an absolute correspondence between the conditions which cause activity of muscle and nerve, and those which capsize what we have chosen to call activity of streaming plant protoplasm. Compare also Horman 98. We might show, moreover, that other types of protoplasm are also acted upon in somewhat similar fascination. When a MXXL cell responds to a stimulus, it contracts. But for the IC-actual type of cell, practically the only type of physical response possible is division. Various agents, Horman 98 has developed a similar concept. For this worker, electrical stimulation, Aragon, of streaming plant protoplasm results in a cessation of movement. Here, too, the term stimulation is not used in the sense in which it is ordinarily used by students of protoplasmic streaming. Because cells to divide, electric currents and mechanical forces are not the best means, but they have sometimes proven effective. See, for example, Lilly and Cattell, 25. Or we might take the case of amoeba. Its normal state is a state of motion in which the cell has various pseudopodia extended. Electric currents and mechanical pressure or shock cause a retraction of pseudopodia. To anyone who has surveyed the whole field, there can hold no question but that in many types of protoplasm, mechanical pressure and electric currents, and some other agents which WC have not chosen to consider, cause a definite activity or RC tree of the protoplasm, and this response is generally prevented by low concentrations of ether or other fat solvent. The point we should now like to make is that in all these types of protoplasm, the effect of the electric current and the mechanical ineosemium on the one hand and of the fat solvent on the other is the same. To the student of general physiology, this may seem almost a truism. To the reader of this hook, it must be obvious that many types of living substance, the electric current and fat solvents, have been found to produce the same effect. In the one case or the other, wherever this effect has been tested, in an earlier chapter, it was shown that electric currents produce gelation or coagulation, or, if one pleases, a great increase in protoplasmic viscosity in amoeba intium descantia. In Pliasolus and Camarmioamid in man, mechanical irisure or injury causes a similar Chang3 in the protoplasm of Vita, Phaseolus, Sical, Jararafanus, Triscantia, and Arbata. Low concentrations of ether were found to have the opposite effect, that is to say, a decrease in protoplasmic viscosity. In Vita Galuzia, Peticularia, 8, Pyrogyrosigulodia, Arbaca. The evidence as far as it goes is concordant. When Yevsirtia action of mechanical pressure or injury has been tested, or whatever the action of an electric current has been tested, botley of those agencies have been found to produce a great increase in teleoviscosity of the protoplasm. And whenever the action of dilute concentrations of fat solvents has been tested, it has been found that they cause a decrease in protoplasmic viscosity. The reader, by this time, will probably perceive whither the argument leads. It is our aim to attempt an explanation of the more complex and less readily observable forms of protoplasmic activity on the basis of what is known for the simpler types of activity. This may seem a logical procedure, but it is against all precedent. Heretofore, the physiology of muscle and nerve has usually been studied and explained primarily by men trained in medical physiology. These workers are, for the most part, not very sympathetic with the protoplasm concept, and these have little or no interest for morphological study. But if it is true that muscle and nerve in their behavior toward various agents are exactly like various other, more simple types of protoplasm, it can scarcely be doubted that the study of these simpler types of protoplasm will yield results of importance for the understanding of muscle and nerve physiology. Objection may be made, and with justice, that the protoplasm of muscle and of nerve is very different from the more simile types of living substance on which our viscosity data are based. This we must admit. But on the other hand, in the case of muscle, for example, it has never been proven that the contractility of the muscle protoplasm depends on the fibrils or striations that can be seen in it. It is probably true that all muscle fibers contain fibrils, and it is only logical to suppose that these fibrils are responsible for the contractility. Against this point of view is the fact that in its behavior toward the electric current, toward mechanical pressure, and toward various other stimulating agents, muscle protoplasm reacts in exactly the same way that the simpler types of protoplasm do, and it reacts in the same way toward ether. In view of these facts, we should like to present as an alternative point of view that the fibrils in muscle protoplasm are not primarily concerned with the process of contraction. This we realize is counter to the opinion of practically all authorities on muscle physiology, present and past. But in view of the fact that muscle physiologists, after so many years of intensive study, have failed to present a plausible theory of muscular contraction from a physical or colloid chemical standpoint based on the presence of fibrils, it is, after all, at least permissible to entertain the notion that the fibrils are not primarily responsible for the initiation of the contraction. Moreover, there is some direct evidence in support of this view. As every student of embryology knows, the heart of the chick begins to beat early in the second day of incubation. The muscle cells which cause this beat have no fibrils. Fibrils can only be demonstrated in these cells on the third day of incubation. This was shown by Eocod 67 and later by Kierulgi 7. Mabsici 3 showed in the embryos of the lizard Anguis fragilis that at a time when the heart beat clearly, no fibrils could be fraud tadpole which moved rapidly and must therefore have had a calculation. Mabjot was also unable to detect any fibrils in the cells of the heart. It is to be noted that Mabjot used elaborate modern histological methods. We may therefore regard it as proven that in vertebrate embryos, generally the muscle cells of the heart can function without fibrils. Similar results have been obtained by the students of tissue culture. See, for example, Lewis 23a. It might be argued that fibrils are really present in these muscle cells, but that they are invisible. This is hardly a valid argument, for it is the presence of visible fibrils that we are denied. If ultramicroscopic fibrils were present, this would be a different matter from a physical point of view. Even though we present the view that fibrils do not originate the muscular contraction, it must not be thought that we deny that they play a part in the dynamics of the muscle cell in which they are found. It is perhaps true that in some muscle cells there are fibrils which by their elasticity cause an expansion following contraction. It is also possible that in some cells there are fibrils which exert a tension on the cell and tend to pull it to get leer, that is to say, to contract it. The presence of such fibrils would not in the least interfere with our generate scheme of interpretation. A muscle cell is doubtless acted upon by various forces, some of which tend to make it longer and some shorter. We can readily conceive that among the forces which tend to shorten the cell may be the tension exerted by fibrils. What we should like to deny is that this fibrillar tension is a rapidly variable factor in ordinary muscular contraction, or to be specific, that it can be made to develop suddenly by an electric current or by mechanical pressure. It seems a fair assumption that the activity or the contraction of a muscle cell without fibrils depends on the same physical principles that it does in a muscle cell with fibrils. If we conceive of such a muscle cell without fibrils as being exposed to an electric current of sufficient strength to induce contraction, it would be hard to believe that the protoplasm of this cell would not react to the electric current in the same manner that all other types of protoplasm which have been investigated have been found to react. And we believe also that the behavior of such a cell toward ether would not differ from the behavior of all the other types of cells that have been studied. An actual test of these statements would probably not be very hard to make. Lewis 23a has described the presence of Brownian movement in smoothly and striated muscle cells in tissue culture. It would only be necessary to send an electric current through these cells and to determine whether or not the amplitude of the Brownian movement was affected. Similarly, one could study the action of ether and other fat solvents. It should be stated that Biedebman 9 has offered some interesting evidence in support of the view that contraction of a muscle cell is accompanied by a coagulation of its protoplasm. Biedebman states that in the transparent muscle of some invertebrates, there is a wave of opacity which follows along the course of the contraction. Thus, the retractor muscles of Sippen endus nucleus are a clear transparent blue in the resting condition, but at the least sign of contraction, the contracted region becomes white and opaque. He records similar changes in snail muscles. In present-day muscle physiology, there is no plausible explanation of how an electric current or mechanical pressure can cause muscular contraction by acting on the fibrils of muscle cells. For a time, there did seem to be a plausible explanation. Engelmann found that a violin string shortened in the presence of AF acid. Following this experiment, it was commonly believed by many physiologists that the production of acid in muscle cells led to a shortening of fibrils, in the same way that acid led to the shortening of a violin string. This view has now been generally abandoned for a number of reasons, chief among which is the fact that the violin string shortens only if it is composed of twisted fibers. The individual fibers do not shorten, see B-Inspect 15. We can scarcely assume that muscle fibrils are composed of twisted elements. Mechanical pressure and the electric current do actually cause coagulation or gelation in many types of protoplasm. If they have the same effect on the muscle protoplasm, we are in a way toward the understanding of the physical aspects of muscular contraction. On ordinary proteins or on inanimate colloids generally, neither mechanical pressure nor an instantaneous electric current has any coagulative action. How then shall we interpret the coagulative action that these agents have on living protoplasm? It has already been brought forward in chapters 8 and 14 that the electric current frequently causes the appearance of vacuoles both in plant and animal protoplasm. Mechanical pressure was found to have the same effect. To anyone who has followed the discussion in chapter 14, there is good evidence that the coagulative or gelatinizing action of the electric current depends primarily on the reaction which we have found of so frequent occurrence in living systems, and which shows certain resemblances to blood clotting. This reaction, it will be remembered, depends first on the presence of free calcium. The calcium unites with some substance which may be found in granules, and a thrombin-like compound is the result. This thrombin-like compound then produces a coagulation which frequently takes the form of films or vacuoles, but does not necessarily take this form. When pigment is present, the reaction is accompanied by a loss of this pigment. There is actually some evidence that the reaction that we have just outlined can occur, and does occur. The evidence is perhaps not very good. No one has ever studied the p-problem of muscular contraction from this standpoint, and we shall have to depend on one or two chance observations in the literature. We shall take up first some relatively recent studies of RSE, Lily on Arenicola larvae, and then some old observations of Kithne. In a series of studies of the larva of the annelid Auricola RSI, Lily 9, Tib, T2, T3 showed that violent contractions of the musculature were always accompanied by the loss of a yellow pigment. Lilly himself saw the resemblance between this phenomenon and the cytolysis, so often described for sea urchin eggs, see Lilly T2, P380, footnote. But for Lilly, cytolysis and loss of pigment were all manifestations of an increase in permeability of the plasma membrane. That loss of pigment has nothing to do with changes in permeability is certainly true in the case of the Arbacia egg, where the process of pigment release from the pigment granule can be studied outside of the cell in the total absence of any permeable or semipermeable membrane. This is a point that has already been considered, CP246. What we wish to emphasize here is that Lilly found in the muscle contraction of worm larvae evidence in muscle cells of the reaction which occurs so widely in other types of protoplasm. In the presence of fat solvents in anesthetic concentration, the muscle cells did not contract, and no pigment was given off. Lilly attributed the action of the anesthetic to an effect on the plasma membrane. But it can be shown in the case of the sea urchin egg that low concentrations of ether tend to prevent an escape of pigment from Tli pigment granule when this is outside of the egg and not surrounded by any plasma membrane, unpublished experiments. However, we must not lose track of the main point that in the contraction of at least one type of muscle cell, there is evidence that a reaction occurs exactly comparable with the reaction which we have described in Chapter 14. The observations of Kithne T4 are even more interesting. Kuhne describes the pre-aeration of a muscle piapisma from frog muscle fibers, and this muscle plasma outside of the cell was found to give a reaction which might easily be interpreted as being akin to tiliate found in the protoplasm of intact coals. In order to appreciate the significance of KTI-ME's ABS zero observations, it will be necessary to describe his methods of work carefully. Ordinarily, if one cuts or squeezes a muscle fiber, no fluid emerges, or if fluid emerges, it travels only a very short distance before a pronounced surface precipitation reaction sets in. In order to res oxidize muscle plasma, KTI worked under the very conditions which would prevent a surface precipitation reaction. From the discussion in Chapter 13, these conditions are known to be: first, low temperature; second, the absence of calcium; and third, the absence of thrombin-like substances. In describing his experiments, Kidimi states, "Schneidet man die Muskeln der Unterschenkel von 8 to 12 durch Injection IT 1%iger Kochsalzlosweg von bindgereinigten Wasserfroschen einzeln. Oh no, sie an Oberflächen zu verletzen herunter. So Kain machen sie zu einem kompakten Haufen vereinigt, bei der bezeichneten Temperatur in etwa 3 Stunden in einen zusammenhängenden festen Eisklotz verwandeln." The temperature in question was 7 to 10 degrees C, and the muscles were used with cold knives and ground in a cold mortar. In studying the description of Kuhne's experimental procedure, it is interesting to note first that he worked at very low temperatures. Second, that he washed the muscles free of calcium by injecting the frogs with sodium chloride solution, and thirdly, that he was careful not to injure the muscle. Injury of the muscle might very well have resulted in the production of substances similar to the ovothrombin described in Chapter 13. Note too that there was no blood and hence no thrombin present. All in all, if Kike had known the exact details of the surface precipitation reaction as we have described it, he could not have chosen his methods of work better in order to avoid it. The muscle plasma prepared by Kuhne is fluid in the cold, but forms a coagulum at room temperatures. This is of course to be expected. Sea urchin eggs torn in the cold form a precipitation film as soon as the temperature is raised. If a drop of the muscle plasma is allowed to fall into distilled water, a coagulum forms immediately. This may be due to a reaction like that of protoplasm toward distilled water, but is more probably due to an action of the distilled water on the globulins of the muscle plasma. But for us, the most important experiment of Kuhne's is that described on page 25 of his book. He divided some ground-up muscle ice or plasma into 2 parts. Into one part, he added frozen coagulated blood of the frog, the other part serving as a control. Both samples were filtered at 0 and the time of coagulation compared as the temperature rose. In the presence of blood, a coagulation of the muscle plasma occurred much more rapidly than when the coagulated blood was absent. It is thus evident, if Kuhne's experiment is correct, that the thrombin of frog blood causes, or at least hastens, the coagulation of the muscle plasma. So too Kuhne interpreted his experiment. Thus, apparently, in the protoplasm of the frog's muscle, there is a material whose coagulation is favored by the presence of thrombin. If this experiment of Kuhne's can be repeated, it is very strong evidence that in muscle, a reaction may occur which is similar to the reaction which occurs during the coagulation of blood. It should be noted that Kuhne's experiment was made at a time when the importance of thrombin in blood coagulation had already been demonstrated, but when it was still not known that calcium salts played any part in the process. If Kuhne had known of the role of calcium salts in blood coagulation, he would certainly have studied the behavior of oxalated muscle plasma. It would be very interesting to FMD out if calcium salts play any part in the coagulation of muscle plasma. But even as it stands, Kuhne's experiment shows that muscle protoplasm, that is to say the sarcoplasm, can undergo a reaction like that described in Chap 14, a reaction which we have shown to be a very wide occurrence in both plant and animal cells. There are three very important aspects of protoplasmic activity. One is that protoplasm is made active by electric currents, by mechanical pressure or injury, and by various other so-called stimuli. It has been shown that the electric current and mechanical injury both cause a coagulation of the protoplasm, and there are reasons for believing that the coagulation is due to the type of reaction we have so often considered. Secondly, fat solvents in dilute concentration prevent protoplasmic activity without killing the protoplasm. This effect of fat solvents has been shown to be related to the colloid chemical behavior of the protoplasm in the presence of these reagents. Moreover, as has been mentioned previously, experiments are through progress which may throw some light on the manner in which the fat solvent produces its effect in those cells in which there is an easily observable internal precipitated reaction. A third aspect of protoplasmic activity, and one we have not as yet considered at all, is the fact that whenever protoplasm is made active, the effect tends to be transmitted from one part of a cell to another, and from one cell to another. This is an extremely important aspect of protoplasmic activity, and we shall therefore attempt to find whether our point of view offers any clues as to the possible mechanism of the transmission of activity. The subject of transmission of activity has been studied mostly in nerves. In SCI of the fact that very many investigators have attempted to understand the transmission of the nervous impulse, it must be admitted that our present theories are scarcely very convincing. They rest almost entirely on analogy, the transmission of the nervous impulse is compared with one or another phenomenon in the inanimate world. To the student of the colloid chemistry of protoplasm, this use of analogy must seem of somewhat doubtful value. It so happens that in spite of many attempts of various workers to draw conclusions regarding protoplasm from the behavior of gelatin gels and other materials supposedly similar to the living substance, this method of attack has never yielded a single truth which could be confirmed when the protoplasm itself was studied. Perhaps the present-day theories of the transmission of the nerve impulse will prove to be true, but at present they are little more than guesses. It would hardly be worth our while to consider every type of protoplasmic transmission from the standpoint of the colloid chemistry of protoplasm. We shall discuss chiefly the transmission of activity in plant protoplasm. It has long been known that when a plant tissue is cut, there is a reaction or change that travels out from the surface of the wound. Actually, there are three aspects of this phenomenon. In the first place, it was noticed that the nuclei in cells near the wound moved in a direction toward the wounded surface. See Tangle 84, Neslet. 98, Nemec 1, Cabling 26. And this movement passed as a wave out from the wounded surface. Secondly, there is a coagulation wave, or at any rate, a wave of increased protoplasmic viscosity that travels out from the cut surface to the neighboring cells. See 1 in 26a. DC also P128. Thirdly, as shown clearly by Nembo 1, there is a wave of vacuolization. In the time following the cut or wound, the zone in which the protoplasm shows vacuolization phenomena becomes progressively greater. Doubtless, the movement of nuclei, the coagulation, and the vacuole formation are all different aspects of the same reaction which travels out from the cut surface. The evidence is strong that this reaction is the same as the one defined in Chapter 14. The fact that there is a marked increase in protoplasmic viscosity, that there is moreover a vacuolization, is certainly a strong indication that this is the case. We have no desire to generalize from this one instance. We should like to point out, however, that if protoplasmic activity does really depend on the type of reaction which we have postulated, then there is a conceivable mechanism for the transmission of protoplasmic activity from one part of a cell to another, or from one cell to another, It will be remembered that the reaction occurs in 3 stages, of which the first is the liberation of free to form a thrombin-like substance, which we have called ovothrombin in the ease of the sea urchin egg, and the third, the reaction between this thrombin H key substance and another substance to form a precipitation product. The ovothrombin of the sea urchin egg was found to be surface active. Hence, it might travel very rapidly over surfaces, F or Brickmack and St. Georgie. 2020 have shown that surface active substances can travel over surfaces at the rate of 25 centimeters per second or even faster. More important than this is the fact that there is evidence that the third stage of the reaction sets free calcium, CP227. If this is the case, the reaction may be self-propagating, just as the reaction which travels along a train of gunpowder is self-propagating. It seems probable that the wave of reactions that we have postulated is in one way or another related to the wave of electric negativity, which accompanies the activity in many tixos of living substance. In view of the importance of calcium in these reactions, it is interesting to note that, according to Ebenstein 26, there is no current of action in a frog retina perfused with calcium-free Ringer solution. These currents return when calcium is again added to the perfusing solution. Further, it should be noted that we have frequently stressed the similarity between the reactions which occur when protoxolasm is injured and when it becomes active. On the basis of this similarity, it is easy to understand why the surface of injured and active protoxylation shows the same electrical behavior. In this chapter, an attempt has been made to establish a point of view regarding the usual type of protoplasmic activity. That such a point of view is desirable is indicated by the fact that the same physical agents induce activity in widely different types of living substance, and that this activity may be prevented by a single reagent which may be used in almost the same concentration both for each lot and animal cells. It has certainly been shown that in all those cases in which investigation has been made, the agents which produce activity cause a coagulation, or at any rate, a great viscosity increase imply protoplasm, and that most of the reagents which prevent activity have the oil side effect. From this certain information, it has been argued that in other species of protoplasm, in which as yet no viscosity measurements have been made, activity is also due to coagulation processes and anesthesia, in general to a prevention or reversal of coagulation. Furthermore, an attempt has been made to inquire into the nature of the coagulation process, and some evidence has been gathered which indicates that the reaction involved may be the specific reaction which was considered in detail in Chap 14.